This week, I have the opportunity to connect with Dr. Rebekah Samsonraj, who has just joined University of Arkansas as a tenure-track assistant professor in engineering and biomedical engineering. Rebekah was previously a research faculty at the University of Oregon and completed her postdoc at the Mayo Clinic. Her primary research focus is in developing strategies for safe and efficacious MSC therapies. I'm super excited to have this chance to interview her as we share a common connection of having spent time in Singapore, where she completed her PhD.
Hey Rebekah, it's so great to connect with you today. I'm looking forward to chatting and learning more about your work. So I know you've done quite a bit with MSC characterization, assessing potency and differentiation, and even aging. Wanted to hear more about how you got into this field of research and what or who sort of motivated you to pursue this work.
Okay, great. Thank you, Maya, for this opportunity to get connected with you and to share my experiences. I got into this field as early as an undergrad when I was fascinated by the potential of stem cells while taking courses in cell biology. And then when I went on to graduate school, I had the opportunity to get hands-on experience with cord blood stem cells. That was my first experience with stem cells. And then the turning point was when I started my doctoral research in the lab of Professor Simon Cool and Professor Victor Nurkamp at the Institute of Medical Biology, A-Star, Singapore, where I began working on bone marrow-derived MSCs. I vividly recall my first meeting with Simon when he handed out to me the lab's recently accepted article back in 2010 in Biomaterials and said, "Rebekah, I want you to read this paper. There are huge issues with MSCs. Their in vitro characteristics and the way they perform in the lab do not correlate to how they perform in vivo." And so I was hooked onto this and was determined to solve this problem. And the next few years, as you can expect, I was immersed in MSc research in Simon's lab, where he provided me with the resources, mentorship, and the environment needed for me to succeed in my research. So my motivation came from Simon Cole and Victor Nurkamp.
That's awesome. Yeah, Simon is a wonderful person, and I've had several interactions with him, and he's always so warm, and just really great person to work with. So I'm glad that you were under his research lab to be working with him so closely. That's really nice to hear.
Yep.
Yeah. So let's talk a bit about potency. That's a very hot topic, I would say. A lot of people, ask me all the time about, oh, how do we develop, potency assays for MSCs? And I know you published a paper a few years ago with Simon, and the paper was just around establishing potency criteria for human MSC potency. And one of the things that was discussed in this paper was how there's differences between these high capacity versus low capacity growing MSCs, right? And you found two markers, STRO1, PGGF receptor alpha, that was preferentially expressed in high growth capacity MSCs, right? So how do you think, what do you think are some of the implications of this work, and how do you think they might be serving as good assays for a therapeutic application?
Yeah, happy to talk more about that. So I started off with the goal to identify correlates and criteria for MSC potency, and we adopted a method of characterization that extended beyond the standard ISCT criteria, which included cell size, telomere length, secretome analysis, STRO1 expression, PDGF receptor alpha, beta expression, and a range of assays. And the parameters that we developed are still being adopted by many labs. And then this work, as you might know, was published in Stem Cells and was listed as one of the most influential articles in Stem Cells, and as well as the most cited. And the strategy that we adopted was to first begin with an ectopic bone formation assay as a simple functional model. And we correlated efficacy in vivo to in vitro functionality. And so we did several correlates, and one of the most important correlations that was almost about, how do I say, 0.95 R squared value was coming up with the STRO1 levels and the PDGF receptor alpha levels, and how people can use this in the future down the line for a variety of assays is to be able to do a simple screening as early as passage 1 in culture, and to get a feel for the percentage of STRO1 positive cells and PDGF receptor alpha positive cells. And so at that time, this was a pretty crude assay. We just didn't want to go with osteogenic, adipogenic, chondrogenic differentiation. It was not just about checking the boxes, but it was more of, can we find out more? Can we extend it? But this was further developed in the next study, the GSTT1 study, where we could find out a marker which is like a yes or no answer. And the strong correlates for bone formation or an efficacy. I mean, we could have easily chosen immunosuppression as a potential functional readout, but we chose bone formation because we are a bone lab. We focus on musculoskeletal regeneration. And so ectopic bone formation was the closest model that we could start off first, because we wanted to prove a way to correlate potency. And therefore the ectopic bone formation assay was developed, and we conducted studies with that. And the main dividing factor was cells that had higher population doubling versus cells that did not. And then we correlated between parameters in vitro and also between in vivo and in vitro. And that gave us very strong, robust correlations. First, the very first factor was growth capacity. Everything was correlating back to growth capacity. If you took STRO1 levels, it correlated to growth. If you took telomere lengths, it correlated to growth capacity. And so we could have easily said STRO1 positive or STRO1 negative cells, but that was not our goal. We wanted something that the community can use, the scientific community can easily adopt, and growth is one particular feature that all labs would test, right? Anybody, whenever we're working with cells, we want to make sure that their doubling times and their CPDLs are all listed out. We kind of document it so rigorously. And so I think that would be one piece of data that any lab could do throughout the world, instead of working with markers or fiddling with DNA and RNA. And so the goal was to make it as easily applicable as possible across the MSC community. And so, and hence we developed that. And so I would call growth as the more qualitative and quantitative indicator of potency. And STRO1 and PDGF receptor alpha levels, so on and so forth, are kind of the parallels of it, which just complete the story.
Sure. I see. And there were criteria. What were the criteria that you set for in terms of growth capacity? Were they different for different tissue? For example, adipose versus bone marrow. Do you think that will be different criteria that we need to set?
That's a good question. Yes, it would be important to tailor it to the tissue source, because adipose MSCs are naturally much faster growing than bone marrow-derived MSCs, based on many years of handling bone marrow-derived MSCs and adipose-derived MSCs. And it would be good to have an in-house metric for specific MSCs from specific sources. But the blueprint remains the same. So you want to be able to get a CPDL of a particular value between P4 to P6, for example, and then you would know that this is a promising candidate. It is a good to go candidate for therapy. You could work on that, and so on and so forth.
Was it a surprise for you guys when you found the GSTT1 that you found a single marker? Was that sort of a surprise, or was that something that you were hoping for?
No, we had absolutely no idea what we were doing. So this work actually stemmed from my 2015 paper, and around the time that I was submitting my thesis, we collaborated with the Genome Institute of Singapore, GIS, with Larry Stanton, and he was really interested in my doctoral work. And then he said, I think we should start looking at the transcriptomic profile of these cells, because now that you've grouped them into high and low, clearly there should be one or two clues that are hiding within these two big cohorts of cells. And
Right.
so let's look into what they are. And so we then we approached Larry, and then he actually um, put together a little plan for us. And so when we were looking at the global transcriptomic profile, it revealed that the high growth capacity MSCs were characterized by a loss of the gene encoding GSTT1, glutathione S transferase theta 1. And then these GSTT1 null MSCs were the ones that demonstrated increased proliferative rates, increased clonogenic and multilineage differentiation potential, longer telomeres compared to the low growth capacity MSCs, which was really correlative. And the low growth capacity MSCs were GSTT1 positive. And so
Wow.
this study allowed us to identify GSTT1 as a novel genomic DNA biomarker for MSC potency. And so it kind of helped us provide a screen for the selection of donors prior to bone marrow harvesting and MSC isolation. And one of the most important aspects of this is that it may be possible to use GSTD1 status as a rapid assessment tool in order to make a yes or no decision for bone marrow harvesting, for example, right? And then when you combine this strategy of selection with MSC bioprocessing and microcarriers, media formulation, so on and so forth, it's only going to help scalable biomanufacturing possible, because you're starting off with a good source. You're not trying to scale up a poorly growing donor with MSCs that's only so much, but you are kind of selecting for the good ones up front, and then you're going to be focusing on producing billions of best-in-class MSCs that have high therapeutic potency.
Right, right. Now, a lot of people also, kind of tweak or play around with culture conditions and even priming steps and so forth. How would that change everything? Would that, change all the expression levels of different markers, and you have to kind of restart and evaluate every single condition? Or what do you think about that?
Any deleterious manipulation to the cells is not recommended, as you would agree, and which is why I think preconditioning cells with minimally manipulative agents like heparin sulfates, which we've also recently shown in stem cell reports as a glycosaminoglycan mediated, minimally manipulative way to enrich MSCs to act better like chondrocytes. So we're basically harnessing the autocrine mechanism. So you're putting in some glycosaminoglycans and you're regulating the FGF signaling and you're regulating secretome, and hence you're making the MSCs behave better in an environment that you would prefer them to work better. Like, say, for example, in cartilage, you want them to function more like chondrocytes. And so kind of tailoring it to the application would be important instead of doing a blanket preconditioning, because the way that you might want an MSC to work for neural repair might not be the way that you might want it to function for bone repair. And so there are several ways to do it. However, given that any preconditioning may not have lasting effects down the line, it's as much as possible to keep MSCs in their naive state would be great. Preconditioning does tend to push them towards a lineage commitment, which may be favorable sometimes, but not always. However, with the proper FDA regulations and clearances, I'm sure that will one day be a possibility. And then with chemically defined media options, going serum free, there are so many aspects to it. And I mean, for us, the goal is to be able to get a cell that will do well in a universal media condition.
Yeah, that's the challenge, definitely.
Yes, it
So,
is.
yeah. So there's also, obviously, as you know, a lot of interest with MSC EVs and using them as therapeutic agents. So what, with your experience in MSCs and developing potency assays for these, what would be your kind of recommendations in establishing potency assays for MSC EVs?
Yeah, great question. Sure, for EVs, you might want to look into ways to profile their protein signatures, kind of determine a molecular protein signature for MSCs, for MSC-derived EVs, because with increasing data and evidence that MSCs function predominantly by what they secrete and not through engraftment or differentiation, you may really benefit from correlating the protein signature of EVs to simple functional assays such as immunomodulation or ectopic bone formation or a simple wound healing assay. And so I would rely heavily on looking at the protein signatures rather than the microRNAs or mRNA profiles of exosomes. And so developing a molecular fingerprint with EVs and maybe a database from multiple donors will give us key clues of what is consistently expressed. And end of the day, we would need to do tremendous amounts of functional in vivo potency correlates. So not just doing in vitro assays may not be the best, but then definitely choosing smaller animal models and having your proteomic profile, high resolution mass spectrometric proteomic profile as the blueprint to which you would then correlate your functional outcomes would be, I think, one of the best insights that I would be willing to offer to whoever is working with EVs, because we need to, because EV cargo is diverse. It's got several things in it, and if there is a way that we could control the secretion, if there are ways without manipulating it to provide uniform secretion, that would be the way to go. However, the field still lacks an atlas of exosome protein profiles. It would be great to develop an atlas, and then we could refer to, okay, for neural specific application, what are the kind of EVs that I want to be harvesting, and which is the best source? Should I go for adipose-derived MSCs? Should I go for cord blood-derived MSCs or bone marrow-derived MSCs? And the field is, I mean, the opportunities are just wide open, and a lot of labs are now working. It would be great if someone's coming up with an atlas, and I'm sure it'll be very useful.
Yeah, I think that's an excellent idea that, if we could pull all the resources to create this atlas, I think that would be very helpful for everyone.
Yeah.
So I know you're joining University of Arkansas pretty soon. What are you most excited about? What are your plans there when you first jump in, I guess?
Yeah, so first of all, I'm excited about this new faculty position and having my own lab, a dream come true, as you would know. My lab would focus on building strategies for MSC-based cellular therapies and biomanufacturing of MSC-derived products using methods that are predominantly biophysical and non-deleterious to the cells. So I've been able to put in place a modulation strategy for scalable biomanufacturing of stem cell-derived therapeutics. It will be heavily focused on the secretum of MSCs, with efforts directed to investigate novel ways to improve EV secretion from MSCs. And the department as a whole it's a highly diverse and collaborative environment with excellent support for research and innovation. And I'm looking forward to this new exciting journey of working with students and collaborators within and outside the University of Arkansas.
Yeah, no, it's definitely an exciting transition for you. So I'm excited for you as well.
Thank you.
Yeah. So you also mentioned last time when we chatted that you're pretty involved with workforce development and creating these training programs for sort of MSC culture, best practices. Tell us a little bit more about what you were doing there, or what your plans are at University of Arkansas as well.
Yeah, that's right. So I'm very much eager to recruit talented students from all backgrounds. And my lab is now hiring graduate students who are keen on engaging in stem cells and cell therapy research. Specifically, I'm looking to recruit students from underrepresented minorities in science and engineering. And one of the most important pieces of my education and research mission is to encourage individuals from diverse backgrounds, including those from groups that are traditionally underrepresented in biomedical fields. And I'm looking forward to mentoring such students and especially investing my time on undergraduate research experiences with the goal to get them interested in pursuing a PhD, as I hope to train a diverse pipeline of scientists for the future biomedical workforce. And so it's more of giving back to the community whatever I received as a minority in STEM. And so these are some of the goals that I have, and I'm open to collaborations with folks because that's been an indispensable component of my research over the last decade. And the idea is to get students engaged in hands-on research and skills development very early on in their education, in their undergraduate education. And so when the time comes for their junior or senior years, they are motivated to pursue a PhD, and then they'll move on to becoming the future scientific leaders in biomedical engineering.
Yeah, I think that's great. I think undergraduate training is so critical in sort of changing the trajectory of a student's direction, right? Especially identifying, students who are eager and keen about research and really into the science, I think, and kind of sparking that interest in them is really important at that stage. So I'm really glad that you're very active in doing that. So that's awesome.
Yes, I'm looking forward to it. And I might knock your doors and send a couple of students to RoosterBio for training.
Sure, definitely. We're more than welcome to, bring students on board as well to train. I think we have, as I mentioned, we definitely are looking into some programs. So definitely looking forward to work with you as well on that.
That's great. Yeah, and I'm sure like our goals of scalable manufacturing and quality control align so well in cell manufacturing, and it would be great to pursue collaborations with industries that have similar vision for cell therapy.
Absolutely, yes. I think the alignment between industry and academia is definitely very important. So, definitely, I think we should have identify some synergistic potential between each other and move forward from there. So, really glad to have this opportunity to chat with you and learn more about your work. So, is there anything else, last words that you want to share with the community before we kind of close the loop here?
Sure, Maya. It's been great to get in touch with you and to also be a faithful customer of RoosterBio, and I've just enjoyed your products. And for me, as an aspiring biomedical scientist and leader in the field, my message is that it's about hard work, and every single discovery makes a difference. Every single result makes a difference. And I'd encourage students, graduate students, postdocs to not give up in this long, hard, painful journey of stem cells and tissue engineering research, and never lose heart, because we are so close to getting our science to clinical implementation. And it's about helping one another. It's about sharing. It's about being selflessly involved, and to have a mind that is immersed or focused on developing real solutions to solve real world problems. So that would be what I would love to share, and I'm available for anyone who wants to collaborate, share ideas, incubate ideas, and so that we could get our science and our lab discoveries closer to the marketplace.
Absolutely. I think patience is definitely much needed in the world of tissue engineering and cell therapy. So, thank you so much, Rebekah, for your time today. And, I think that everyone will be very excited to hear more about your work coming in, and we'll definitely stay in touch.
For sure, Maya. Thank you for the opportunity. Thank you for chatting with me. And I'm here whenever you need me.